Today I will be talking about the Signals Research Suite and specifically its use for biologics. You'll have heard us talk many times in the past about this make, test, decide workflow. And so today there are a couple of things we're going to emphasize. The first is this concept of the Signals Research Suite. And so we've been talking about this, but I really want to drive home what this means for our customers. In years past, we have made a big point about how thoroughly we are able to help our customers manage the make, test, decide process for doing their science and admitting their medicines and products to bring to market. And these best-in-class tools have now been brought together into an integrated, unified platform called Signals Research Suite. Signals Research Suite is the same fantastic capabilities, market-leading capabilities in data capture and data processing, in experimental capture and decision support, but now delivered into a single SaaS package. And the other new thing today that we'll be focusing on is biologics. We've talked for many years about our market-leading world-class capabilities for small molecule discovery. But today we're focusing on another part of the market, that for biologics. So you have heard me talk in the past about this concept of Harper Pharma, which was the kind of exemplar small molecule company that we use for talking about our platform in drug discovery. Well, today I'm going to be talking about Harper Biopharma. This company is also a exemplar company whose focus is on doing antibody-drug conjugate development, where they need to assemble together this complex macromolecule structure that will deliver a payload to a therapeutic target of interest. And like Harper Pharma, they're at Series B, they're got a first program going to the clinic and many other programs underway. So what makes the biologic space more complicated or uniquely challenging? And it really comes down to the fact that the molecules are larger and harder to represent and harder to grapple with. A lot of small drug discovery, of course, happens down here at the small end of the mass scale. And as you grow to peptides and full enzymes and full antibodies and all the way up to cells and stem cells, your representational challenge changes from something you would draw in ChemDraw to something that you need to draw in Helm and beyond. And what we're talking about here are things like peptides, oligos, full proteins, vaccines, and so forth. We have a capability set that extends all the way up to these much larger molecules, but we haven't talked about them as much as we should. I'm going to rectify that today. And the right representation really depends upon the user's goal. If they're focusing on small molecule drug discovery and development, of course, ChemDraw is their tool of choice. It's the best tool for being able to explicitly represent where every atom exists in the structure. And then there's Helm, which is a standard that came out of the PSOE Alliance many years ago. And that's the best way to represent a peptide or an amino acid or an oligo, or a nucleic acid or oligos, where you might have modified side chains, different branching structures, and so forth. But you still want to represent things not at the level of the individual atom, but really at the level of the individual monomers and constituents of that polymer. Or if you get to as large as a full RNA, DNA, or protein sequence, you might want to really think about things in terms of the two-dimensional structure of that amino acid chain or nucleic acid chain, and focus in on regions of interest, like the CDR regions of an antibody. And finally, if you're really doing full protein development, it might be the full protein structure that is what's relevant. And so we've really delivered capabilities that I'm going to span this gamut, and I'm going to be talking about these in the middle, which we haven't spent as much time on as we should have today. So Harper Biopharma is made up of a cast of characters that really drive this process. Alice is the scientist that is involved in antibody discovery. She injects the animals to generate immunogenicity and then harvests the cells and tissues to make the hybridomas. Alex then will contribute to the team by testing those hybridoma clones and the downstream antibodies and antibody-drug conjugates in a variety of assays. Stacy is a specialist that works in SPR, which is a technology stack often used in larger molecule development. Paul will then help by optimizing the protein structure and the expression vectors to be able to mass produce those constituents. And then Charles contributes by being the person who delivers the payload as well as the customized linker that brings all these things together from the antibody to the antibody-drug conjugate. So here's your cast of characters in Hopper Biopharma. And their process, let's talk about the process for a moment. They begin by injecting animals to generate novel antibodies with the correct immunogenicity, and then harvest the cells from and generate hybridomas. They will then test for primary activity, typically in a high throughput screen, typically with technologies like ELISA or other primary screening activities, and also selecting some of the lead hits to go into secondary assays like binding assays and SPR. Once the team has gathered enough information about the antibodies that have been generated, they will go into selecting the antibody to be advanced into ADC, antibody-drug conjugate development. And those lead antibodies will be cloned and have their chains optimized for protein expression and expression systems of a variety of sorts. The antibodies are then expressed, the protein's purified, or rather the antibodies purified, and then these will be combined with the payload and the special linker to make the final antibody-drug conjugate that will go through a battery of assays and on into final antibody-drug conjugate candidate nomination to move on to development at the clinic. So this cartoon simplifies what is a vastly complicated multi-month process that maybe is best represented with a huge flow chart, but for today's talk, I think we'll simplify things a bit. So here's the outline of the rest of the talk. Discovering antibodies of interest, engineering them together with linkers and payloads, checking their binding, tracking the linker development, the linker sequence using Helm, integrating and combining it with the small molecule payload, and then testing the full ADC in some kind of a cell killing assay, a variety of assays to identify the correct one to advance. And of course, finally, sitting down with the full team to decide which antibody-drug conjugate is right. And it all begins with Alice. Alice is, again, the antibody discovery scientist who will work with the animals and go into the vivarium. And Signals makes it easy for Alice to track her overall experiment. In the Signals notebook, she can gather the protocol and the experimentals. She can also begin capturing all the information about the media and the solutions that are being used in the process of this immunogenicity generating experiment, where the animal will be injected with proteins and then monitored for basically immune response. To monitor for immune response, Alice is able to use Signals to gather a plate map and gather information from the data points as the experiment progresses, and analyze these data points in Signals using our Spotfire capabilities to track how the immunogenicity is progressing. Alice really values Signals because it helps her complex workload and her complex day where multiple animal studies are going in parallel. And Signals Research Suite is really designed to make it easy to plan those studies, execute them, and then centralize the data management and analysis. Once the hybridomas have been generated, they get handed over to Alex, who will begin running them through a battery of assays. Again, in Signals, he's able to assemble the media and the experimentals and the equipment that's being used to run a high throughput plate screen, generate the raw results, and bring those raw results into Signals as part of the experiment record. From there, then, Alex will run a Signals vitro vivo experiment. Again, this is using our highly tooled and integrated version of Spotfire that's designed to work with the platform. And going from end to end, because all the information is integrated, it's easy for Alex to track and centralize this data processing. The raw data comes directly in from the Signals notebook page, as well as the experimental properties. The plate map's also brought in, and then the specific experimental information about that plate is integrated in to a final assemblage of data. Alex then uses our HTS QA/QC app to normalize the data and to look for outliers. And then finally, we'll progress this to, I got those in reverse. The normalization is happening now in the typical NPA normalization. And then this proceeds into a final hit selection app that makes it very easy for Alex to identify the antibodies that are most likely to be successful in the next steps. For Alex, the main benefit is it centralizes the data and automates the processing such that standard workflows can be done in a standard fashion. Those results can then be brought back into the platform to be sent downstream for the task of cloning and final sample workup. Oftentimes, though, the scientists want to also verify the binding affinity of the antibodies of interest. And so for this, the antibodies will go to Stacy, who will do an SPR experiment. We have built very thorough capabilities into our Signals platform for being able to process the SPR data from raw results all the way to final product. And as before with Alex, all of the information is integrated, including the raw data, as well as all the experimental properties coming from the Signals notebook page. The raw data for the SPR experiment is brought in, and then the normal process that is used for reducing those data to final kinetics is able to proceed, including zeroing, cropping the results to the defined range where there's values, referencing so that each of the flow cells can be correctly referenced to its reference flow cell for intra-flow cell, intra-data normalization. The normalized data is then blank subtracted, so you can actually correct the levels against the baseline of the experiment. And then finally put into kinetic modeling. In this case, a one-to-one interaction with mass transfer to look for the Ka and Kd coefficients for the antibody against the target. From here, QA/QC to knock in or knock out points, of course, can be done. But most importantly, this mass transfer kinetic is then calculated and can be QA/QC directly from here to identify which of those values and which of those antibodies have results that are of interest to the team. And of course, these results can then be published into the data repository or the data lake after having brought in information about the specific samples. And so this is the publication app that's used throughout the Signal system to bring experimental results into the data repository layer where it can be integrated with other information and then be assessed for which is the best antibody to advance. The key benefit for Stacy is that this integrated platform allows for there to be a standardization and a normalization in the way the SPR data is processed and managed. There are many instruments, many SPR instruments out there, but they all have their own unique idiosyncrasies and their unique challenges in terms of data processing. With Signals, Stacy has a common platform in which this SPR data can be processed in the common way, guaranteeing consistency of results and a single interface that her team has to learn to use, as opposed to many. Having generated the hybridomas, tested them in multiple assays, the team's now ready to decide which of those antibodies is most powerful or most likely to be successful. To do this, they go into their Signals Inventa analysis object. And this is launching a Spotfire-based tool where all of the antibody information, the antibody properties, as well as all of the assay results related to those antibodies are brought together. Here, the scientists are able to look at all the individual results, as well as the different CDR regions for those antibodies. They can pin a single antibody as the lead and then look for patterns of mutational differences between the lead antibody and any of the other antibodies that have been assessed, sorting by a key characteristic of interest, in this case, a HEC titer, and selecting for the lead molecules allows for a very easy review of which positions see differences. But it's easier to see that in a multiple sequence alignment. So moving over now to the MSA app, the user is able to see this high-level overview of the antibody above, again, pin a single reference molecule, and then look for the mutations that occur relative to that reference. This region that the user is sliding around highlights the distribution of the HEC titer results and makes it very, very fast and easy to identify changes that are driving a better HEC titer. But even better is to have the system return to you in an unbiased fashion the best antibodies by CDR region. In this antibody component analysis, the user is able to select those that show an improvement in the assay of interest, select a reference, and then see the common patterns of mutations within the CDR regions that are driving the outcome that the team is looking for their project. Once those best molecules are selected, the next step is, of course, to send them back to the Signals environment for nomination and moving further into the development process and the antibody-drug conjugate assembly. For this, the team selects things, adds them to a group, and that group list is then available directly from here inside of the Signals analysis object, where the individual antibodies and the results can be reviewed directly from here as the team decides how to progress the overall project. For the team, the main benefit is by identifying these driving amino acid substitutions that lead to the optimal antibody efficiency, they're able to more easily identify which molecules to advance. And Signals Research Suite really helps surface those changes by making it easy to integrate all the results into a biologically and biologics sensible and knowledgeable analysis interface that really is designed to make that analysis process as easy and transparent as possible. Having identified the lead molecules, the next thing to do is to optimize their expression. And Paul, the protein chemist, will then go in and do a cloning experiment in which the individual sequences of the antibody chains will be cloned into an expression vector. We've integrated tools like SnapGene to facilitate this process in plasmid design and assembly. Within this, using the great capabilities of this tool and other tools we've integrated, Paul's able to look at the sequence of the final planned plasmid, identify the restriction fragment linkages that will be used to assemble this final plasmid design together. And this is all happening within the context of this Signals record that keeps track of the original plasmid, the primers being used for the insert sequence itself, as well as the final expression vector, as well as tracking the entire experimental process by which that cloning experiment's being done and developing and assembling together the proof that the correct final plasmid was constructed. Once the plasmid's made, it's of course transfected into an expression vector, and within Signals you have the ability to also track the workflow for that transfection and the step-by-step process of the downstream expression, protein purification, and final proof that the right thing has been made is being assembled all together in one place. So for Paul, the benefit really here is, the challenge here is to do the correct plasmid design really requires careful planning and thoughtful assembly of the parts, as well as using best-in-class methodologies like Gibson and Golden Gate to help parallelize and streamline the process. And Signals really makes it easy to hold all that information together and assemble a single cloning record that drives that process from end to end, as well as integrating best-in-class tools like SnapGene that really facilitate the molecular biologist and protein chemist workflow. The next step in the process is that Charles will develop the payload. And as I mentioned before, we've talked in the past many times about our small molecule capability set, so I'm not going to emphasize that here. For those interested, there is a seminar that was given previously, and the QR code to take you to that seminar if you'd like to see it's there. And it summarizes this process whereby Chloe, the chemist, will synthesize molecules and send them over to Alex for assay processing or assays to be run on those small molecules. And then Alex will return the results back to Chloe, and as a team, they'll decide the best payload molecule to advance. Charles, however, is also involved in the development of the linker. So the linker is, for Hopper Biopharma, kind of the special sauce. It is the technology that they're bringing to bear that differentiates them from other companies that are doing antibody-drug conjugate development. So for Paul, what he really needs is the ability to do a synthesis page that represents and is able to correctly diagram this complex linker that is their secret sauce. We've integrated into Signals now a Helm support that allows for these natural and custom monomers to be rapidly assembled into the polymer chain of interest. And these can either use, of course, the naturally occurring amino acids, the Pasteur Alliance monomers, or any custom chemicals or side groups that you as a user might be interested in assembling into your linker or any other polymer like molecule. This is really a differentiating and best-in-class capability to really make it as easy as possible to diagram these novel polymers, be they amino acids or nucleic acids or other types of polymers, in a synthesis reaction that can then be used for the full workup, just like you would in other contexts. The real benefit here for Charles is it makes it really easy for a chemist who's, or a chemical group that's developing these unique custom polymer chains or oligos or CRISPR RNAs or other types of intermediate-sized molecules to manage those custom monomers, to assemble them together in an efficient fashion, and to really drive an efficient synthesis process. Next, Charles and Paul will assemble together the various constituents of the antibody-drug conjugate, an element from the antibody chain, as well as that custom linker, as well as the payload. And this capability to bring it all together and track all the constituents that are going into something like an antibody-drug conjugate is really an incredibly powerful and unique capability that really allows for a company like Hopper to do their final assembly of the antibody-drug conjugates and trace all that lineage and componentry that went into that. The key benefit here being really that full traceability to allow for that final assembly of molecules that really span that gamut in terms of size. Those antibody-drug conjugates will, of course, go into other testing, and then the team gets together to really decide which of these molecules to advance to the clinic or to downstream development. And again, to do this, they go into the invent analysis object, fire up their dashboard and signals, and here they're able to do a query where they're able to pull together the information from the antibody, the linker, the payload, as well as the final antibody-drug conjugate. And that's really one of the key things that's differentiating here, because they're not just pulling in a single entity, but they're pulling in a bunch of related entities. We now support and have for about a year this capability of having a core entity like an antibody-drug conjugate that has a set of related entities, all of which will be assembled together and all of which could be part of the testing regimen. So within this global search query, then the team runs and brings down that data and is able to see a dashboard that brings together information that is on the antibody-drug conjugate itself in the green, on the original antibody in the blue, on the linker, excuse me, the blue is the linker, the brownish here is the antibody, and then the payload tests. So here you're looking at tests that run against a broad variety of the constituents that go into this final antibody-drug conjugate, and to bring all that information together to be able to derive the decision-making as to which of these molecules is the best to advance. As I'm hovering over various bits here, you'll see this form view pop up to really bring that information together into a kind of a coherent snapshot that makes it easy for a user to understand a complex data set. And then they might want to start looking at things like, okay, how does the payload result compare to the antibody-drug conjugate result? But really, that's insufficient. What the team really needs to start thinking about is a multi-parameter optimization. In this view here, each of the individual axes represent that constellation of data I was just describing across the different components and constituents, as well as in the final antibody-drug conjugate. And here, this view shows the distribution of the results across those many different axes and makes it easy for a scientist to see which of those antibody-drug conjugates is most likely to be successful, which in this case is Hopper 4162. This is the payoff, because you've now gone through, within Signals, this entire workflow and challenge of assembling the information together for an antibody-drug conjugate, tracking all the different layers of information you have to track, assembling it all together into a final dashboard that really drives home and clarifies the message as to which of these molecules is most likely to be successful and to make the right decision about which drug to advance. Using Signals, then, this whole process is able to be driven very rapidly, very thoroughly, from end to end. And it is all happening within the Signals Research Suite. This great SaaS-based capability that brings together these market-leading capabilities that have been part of Signals for many years into a single final SaaS platform to make it as easy as possible for your customers to benefit from it. Thank you, and I'll happily take questions.